And I think since we still have like 10 minutes or so left, it would be really good for us to also hear some potential ideas from the audience. So, don't feel shy. And I know from individual conversations during the day that some people have some ideas. Because as we pointed out at the very beginning, these are just ideas. There's no clear commitment of no one yet, just ideas. And we factor in all the input that we have received. And then we will see what we move forward, what we will not move forward, and so forth. So just ideas. And so we start there. Thank you.
Thank you. My name is Samantha Walker. I'm from a patient charity called Asthma UK. One of the most successful previous IMI bids was UBioPred, which Pierre Moulin mentioned yesterday. And we haven't worked up our ideas as comprehensively as those to date, which are very impressive. But I think there's a lot to be learned from the findings of UBioPred, which essentially is trying to work out the biomarkers that can identify the sorts of asthma that people have got. Because at the moment we've got no diagnostic tests to say whether you've got asthma or what sort of asthma you've got. And this is a real challenge because then you can't get people on the right treatment, and we've got new expensive monoclonal antibodies being developed, which then you don't know who they're going to be suitable for. So it's our idea that this is a problem that needs solving between the pharmaceutical industry, who need to be developing products based on these biomarkers, but we haven't got the biomarkers. So there's something around a public-private partnership that's about the industry getting together to say, what's the next phase of work building on UBioPred that enables us to identify those biomarkers, turn those into inexpensive, non-invasive, near-patient diagnostics, enabling you then to be able to target drug development for the next wave of treatments. Because 3 people in the UK die a day of asthma, and about 250,000 people in the UK have the sort of asthma where there are just no therapeutic options and they've got really severe disease. So I think that there's an established unmet need, and it's whether we can get that public-private partnership between academics and the industry and people with asthma to say this is something that you could solve in this kind of way through an IMI.
Okay, thank you very much. So this is something not exactly specific to the space of ATMPs. This is a more general comment that there's a tremendous need for biomarkers in this area. And new treatments per se. Okay, so what does our panel think, for instance, with regards to this idea?
Okay. I think there is a wealth of learnings to be mined in terms of how you've been working successfully across companies and with academics to tackle these challenges, and what I don't know if if you actually moved these biomarkers forward, and if you managed to get them qualified by bringing the regulators on board, which would be the next natural step, and what we would like to have at the outset, moving into the ATMP space, so having a more coordinated approach and oversight by them as well.
I think there's indeed, as Georg was saying, something which is innate into the new therapies. But I think for ATMPs we need still, I guess, to little bit split up, because you have ATMPs which are really about wound repair, which are about replacement of tissues, where, let's say, the classical biomarker research would less be applicable. Also these gene replacements, where in thalassemia, for example, it's quite obvious. On the other end, of course, if you work in rheumatology or others, there will pop up. And we are approaching this, and I think there are two aspects. One aspect is really the biomarkers more related to the disease, but what we see also, and that's where the importance comes from, because the mode of action of these cells can be quite complex and influenced by the patient. We have already seen that you can have some ideas about prediction of what is the setting where the cells will likely elicit a response, and where the eliciting the response is less likely. So it is included in it. I think one of the big challenges is to really validate a biomarker. You need huge populations, and most of these trials are quite limited. So we have indications, but that's the difficulty.
Okay, thank you very much. We will take this up in any case as an idea and basically discuss it further. There's some more ideas, most likely. Thank you.
This is Guerra from the University of Pisa, Italy. I was always puzzled about the fact that many of the therapy for cancer, can really be validated on the spot on the patient. I mean, you never know when you give some some treatment, if it's going to work exactly as you expect or not. And so there is a need to have a fast response. Now, I wonder how the IMI would react to a proposal when an idea tried to making some device that is cost-effective, very practical, and give the response immediately. Of course, I have something in mind that making a molecular imaging device at very low cost specific for this type of investigation. I wonder if could be of interest of the consortium.
Immediate reaction from the panel? Anyone else in the audience, actually?
I don't think we can give a committing response. It's another idea to consider. In terms of requesting the support of a public-private partnership, I guess what's the degree of collaboration that's needed here? If you like, you could clarify, or in order to substantiate a proposal, you need to clarify that this is something that needs more than a player or a few.
But I mean, it could be, if this is a little bit sort of small scale for a really big project, thinking big always, that could perhaps be made a work package in a broader context of a program, actually in IMI. That would be a possibility. It would be a big project per se. What do other people think? Okay, but we take it up as an idea and will be investigated further. Okay, thanks.
I just want to bring up the topic of GMO, genetically modified organism, documentation and review. I think a colleague already mentioned it. And as long as we park it or have it reflected in the clinical development section of one of those 5 topics, I think that's fine. I just want to make it a point that this is important, and the longest review periods, and if you compare ethics committee documentation and review health authorities, the GMO aspects take much longer, are less harmonized, and if we could help to make that more streamlined, that we can go in with more centrally developed documentation that is worth to go for all the different countries to address the requirements, that would be helpful. So some work on the GMO documentation, harmonization and review timelines.
This is a proposal from the preclinical area. I would like to make the proposal to put some resources or create a project for IMI on tissue cross-reactivity for development of CARs. I think that the classical tissue cross-reactivity studies, study designs which are out there, which are used for monoclonal antibodies, even for monoclonal antibodies, they have their downsides and their limitations for the interpretation and also technical limitations. And that is also more than true for at the end for the CARs. But nevertheless, we need to somehow address tissue cross-reactivity of these kind of ATMPs. And I think that would be a good forum or opportunity to have a collaboration on new ideas how to assess that. And the key point is what regulators are expecting is that you provide some data on key tissues and you somehow cover that. And that is at the moment a bit difficult.
Okay, good. Does anyone want to comment on this?
I think it's a very good point, but we should also, if we do something like that, really reflect that this is going to be very product specific and likely also carrier specific, because I mean, for let's say the CD19, you could do this cross-reactivity, but there would be other ones. So I think it's a fair safety question to do, though. But what would be the targets to do so?
Okay, a few more minutes before everybody comes back in again. Yes, please.
as well. ATMPs are not only the somatic and the gene therapy product, also the whole regenerative medicine and also mixed products. And we have mentioned IPS today, but I think this should go into that focus as well. You are probably right. It's a little bit behind other products like gene therapy products, but not so far, so behind that we should not include it. And there are really some good projects and also close to the clinic, or some really early first in man or under hospital exemption trials. So we should focus on that as well. I would like to include that.
I think you're right, and that's also why I think the registry can be important, because the registry can also take up from all clinical trials, even if it's early clinical trials, but these data will be available. I think we should, at the end of the day, we should realize that what is different from classical medication, we come, one, with an extremely complex molecule into the body. This is not a TNF-alpha or a small molecule, so this is tons of antigens and lipids and so on, and it's a living product which we bring in. So this is not a standstill. This product can even evolve within the body at a certain moment. This is big unknowns, but at least we could focus on the readouts on what is the body doing against such new type of complex living drugs.
Can I add something to that? Yes, you are very, very right, actually. I think what we're tackling, what we're dealing with here is a lot of variability. There is variability across the ATMPs group, if you will. There is variability across different patient administrations and between product batches as well. So what I think has been a huge gain here is to start identifying and understanding what are the gains that collaboration can bring to that space, and also through the day build that collective understanding that we need to look more coordinately across the end-to-end, essentially, in product delivery. And with that framework in mind, we can perhaps identify the types of projects where work streams can differentiate between cell or gene or regenerative medicine products and provide some structure in all this variability, essentially.
So thank you very much. This will be taken into consideration, for sure it will. And I think now we come to the last, let's say, comment or idea, hopefully an idea, before we close. Please.
Okay, I'll put it as an idea then. I'm Stefan van der Spiegel from the Commission. I'm a colleague responsible for the transplant sector, so that's organs, but also bone marrow and other fields. And I just wanted to offer a bit that many of the topics you bring forward here as issues like immunogenicity, data collection, follow-up, there is already quite some expertise with that in the transplant communities, both at authorities level, but also associations like EBMT. And it might be quite interesting to learn from that, again, not to reinvent the wheel. And besides, of course, that starting materials for the whole ATMP sector come through the tissue establishments. So I just wanted to offer that as an additional tool or area to leverage for your work.
That's great. Offer gratefully accepted. Nobody wants to reinvent the wheel in IMR. We want to capitalize on what exists, and that is why we need to know it. So thanks for bringing this to our attention.